This is CRISPR automated reasoning. Some background that's necessary to know before explaining the project is some biology. So a genome is basically a string that's 6.5 billion characters long that hold our genetic instructions. Recent advances in gene editing has shown that 2% of these create the largest effects, and we're now able to just look at this part, which is called the exome. Now we can introduce the problem. So editing some genes are harder than others. So let's take gene 1 here. There's an issue with gene 1 where it's overactive, which has the side effect of a disease. The problem with editing gene 1 is that there are many copies of this gene in the exome, the important part of the genome, and it isn't feasible to edit all of them. This is one of the many problems that biologists have with gene editing, as some genes are just harder to deal with. This leads CRISPR to not be an economical means to solving certain genetic problems, and this will certainly be a roadblock as we attempt to cure genetic disease. But if you can't get through the door, try the window. Let's say that there's gene 2, where increasing gene 2 decreases gene 1. If we can enable an increase in production for gene 2, this will indirectly have the desired effect of decreasing gene 1. Often, this can get more complicated. Of course, through logical deduction and information, a human can reason through relationships between certain genes to find indirect ways to get desired results. This obviously also isn't a sustainable task for a human, as the exome sequence is about 180,000 characters.
So without further ado, here's a demonstration of the CRISPR automated reasoning system. We can see in line 38 that we've defined that an overactive gene 3 causes Huntington disease. So we're going to use the may treat predicate to see what variable x may treat Huntington's disease. After the query, we can see that the model is quite complicated. This is because the logic tree must go through many trees in order to reason out the justification. So here we can see that gene 5 may be able to treat Huntington's disease. This is because an overactive gene 3 causes Huntington's disease and holds for gene 5 and gene 3. So because gene 5 decreases the overactive gene 3, it may be able to treat Huntington's disease. This is one application that can be used with a given disease and a variable x. The next predicate I'll demonstrate is the also decreases predicate. We can see that we've defined some facts about certain drugs and genes decreasing each other, but these are only length of 1. The also decreases will allow us to see other links. So we can see that gene 1 also decreases gene 2, even though they aren't explicitly defined anywhere, because gene 1 increases gene 3, and gene 3 decreases gene 2. Therefore, the system can reason that gene 1 will also decrease gene 2, even though that relationship is not explicitly defined. This is a nice extension and will allow the system to reason out different facts like this without the human having to do any work. Finally, we'll take a look at editability of genes. We can see here that gene 1 is defined as an essential gene, but we don't necessarily know whether or not it's editable. So if we go here, we'll go ahead and paste this query in. We'll see if gene 1 isn't editable. So it turns out that it is not. It's not the case that editable holds, because gene 1 is an essential gene, and we've predefined a rule that says an essential gene is not editable. So the computer can also reason out information like this without the human having to input anything. The CRISPR automated reasoning system is able to find workarounds to uneditable genes. It's more economical than large-scale clinical trials, because the much smaller trial size helps treat rare diseases more effectively. The continuously evolving database and automated reasoning that can infer other relationships also makes it a very powerful tool for CRISPR research. As we've seen, the database not only stores facts, but is able to abstract other facts using rules that have been defined. The automatic reasoning can also be extended to millions of relationships, which allows CRISPR to be a more cost-effective option. Finally, why is this useful? Well, as NLP capabilities improve, more sophisticated NLP can create huge datasets of relations from reading medical publishings. And as CRISPR improves, genetic research will require such large datasets of relationships so that it can better understand what the gene that they're editing will affect. We're excited to see where the future of CRISPR research takes us, and we're excited to see what tools can be used to implement it. Thank you.